In the second part of this lecture, we want to talk about synaptic vesicle recycling, the endocytosis process that has to happen in order to maintain. Now, we've already looked at the Heuser-Reis model of full collapse and fusion, where vesicles, after dumping their contents, merge fully with the plasma membrane and then are endocytosed either in vesicles or large endosomal structures, from which new vesicles can then be reformed and move then into a storage pool ready for movement to the plasma membrane and fusion again. But there's lots of different versions of this kind of recycling. Does the vesicle membrane stay intact, or does it break up into lots of little pieces and fully merge with the lipid and proteins in the presynaptic nerve membrane? If that's the case, then in the endosome there has to be a lot of sorting to congregate synaptic vesicle proteins in the portion that will then bud off to become synaptic vesicles. A much more efficient possibility for maintaining vesicle integrity would be for, after vesicles have opened and dumped their contents, that they close rapidly, relatively close to the place where they fused, without having the vesicular contents and lipids merge with the plasma membrane. And this has come to be known as kiss and run, well, where the vesicle opens a fusion pore, releases neurotransmitter, and then quickly leaves the presynaptic nerve membrane. It's also been called kiss and stay, so that it stays nearby, ready and competent to fuse again. So the recycling from here may not actually be to a storage pool, but may be back and forth between release and replenish, release and replenish. And then there's all kinds of different variations on this, ones that exclude the endosomal compartment, etc. So one has to think about this mechanistically very precisely in order to look at this endocytosis process. What are the molecular steps that become involved in the full collapse and fusion? We, of course, have found lots of proteins to be involved in this highly regulated process, just as we have for vesicle fusion. And let's go through a few of those. After full membrane or vesicular membrane fusion to the plasma membrane, in order to invaginate, we need what's called nucleation sites, places to tell the intracellular machinery this is where it's appropriate to begin to form a concavity and pull membrane back into the cytoplasm. And the proteins that act as nucleators are ones that attach to different adapter proteins called adaptins. There's a number of these here. Synaptogenin, AP2, AP180 are some of the major ones. And those adaptins then bind to clathrin. And we'll talk about clathrin. But what is the nucleation protein? Well, surprisingly, that's our old friend synaptotagmin. Now, synaptotagmin has those C2a and C2b domains which bind our major intracellular regulator, cytosolic calcium. So that lends the idea immediately that perhaps this endocytotic process is going to be regulated by calcium as well. Oh no, yes, another role for cytosolic calcium. Once the clathrin begins to form, and we'll see from its molecular structure how it begins to pull membrane in and invaginate, it forms almost a fully spherical vesicle, but again there's an energy barrier that has to be broken to pinch the vesicle off from the plasma membrane, and there we get another protein that kind of chokes off this stalk, and the choking protein is called dynamin or dynamin. If you're a neuroscientist, we pronounce it dynamin. If you're a cell biologist, it's pronounced dynamin. But dynamin or dynamin is both GTP dependent, it's IP3 dependent, and basically it provides a cinching action, like a belt tightening, to actually pinch off the vesicle. Then the clathrin-coated vesicle moves through the cytoplasm. Clathrin has to be uncoated in order for this newly pinched off vesicle to fuse into an endosome, where protein segregation of appropriate synaptic vesicle-associated proteins would take place. And then it has to bud off of the endosome, again through a clathrin mechanism, in order to form a fully competent synaptic vesicle, which then, of course, still needs to be filled with neurotransmitter and stored back in a storage pool. This is all quite complicated and requires ATP or energy dynamin stage and in the uncoating of the clathrin stage.